Okay, it's our last session of the conference. And so for this last session, we're talking about the GRI phenotype. And we have speakers who are presenting. First two speakers, Dr. Tim Benke and Dr. Jennifer Bain, are going to talk about GRIN disorders. And then Dr. Allan Bayat is going to talk about GRIA disorders. So I want to welcome our first speaker, Dr. Tim Benke. Don't sit down. From Colorado Children's Hospital.
Thanks, everybody. Yeah, I guess this session is what's between you and heading out, dinner. Thanks very much to the organizers for asking us to present today. And so I think I'm a bit of an overview of things, and I apologize to those that are speaking after that if there's some overlap as we go through this. So I'm going to talk about GRI phenotypes and just as in really terms of a general overview. So these are my disclosures, nothing that's really super relevant to what we're talking about. These are my acknowledgements. I have a lot of great people to work with. The group at Emory, Steve as the lead there that's keeping us all funded for doing this. But of course, in the past, we've had funding from GRIN 2B as well as Citizens United for Research in Epilepsy. This is my group at University of Colorado and Children's Hospital Colorado, Johannes's group in Leipzig, Jennifer at Columbia, the Cleveland group, and the UCSF group that's all part of Steve's grant. So I'm not telling you anything you don't know, but just to say it very distinctly, is that GRI, neurodevelopmental disorders, they're all unique and they're well-defined syndromes. And of course, as you know, that GRI stands for glutamate receptor ion channel, and it's responsible for almost all excitatory neurotransmission in the nervous system. There's 4 types. There's GRINs, there's GRIA, there's GRICs, and there's GRIDs. Each type contains 4, sometimes 5 subunits. Each subunit is a different gene on a different chromosome. There are genetic alterations of any gene cause a different neurodevelopmental syndrome, and gain or loss of functions, our theory is that it causes a different subsyndrome within that. So there are likely then also to be specific therapies for each subsyndrome. And basically you end up with at least 9 different syndromes for each of the different disorders. And what's emerging are these things called grid disorders. These are channels that don't necessarily form ion channels, but they're responsible for synaptic integrity. So GRI disorders, they're common neurodevelopmental disorders. We know from Johannes's work with regards to birth incidence that these are pretty common when you think about how they compare to other common neurodevelopmental syndromes like Rett syndrome or Dravet syndrome. And, you know, their clinical trials have been underway. But one of the big problems is that there are no specific ICD-10 codes for any of these disorders. So let me just make sure that you understand what that means is that basically if you see your physicians in this country, they will code you for what you're being seen for. Did you know that if you go into the emergency room because you were on a scooter, one of those crazy scooters that you can rent all over the place, you fall off and you hit your head, there's a specific ICD-10 code for that. But there is none for us. I have a problem with that. Do you have a problem with that? Yes. So what that means is that we can't ask basic questions about what is the impact of any of these disorders because we don't have the tools there to do it. There was a group of us that recently went through the motions of jumping through small little hoops to see if we could get the CDC on the same page with us such that each of the syndromes could have their own specific ICD-10 code. Now you're going to go in Europe, but we have ICD-11. Everybody has their own code here. And it's like, well, we wish that we would move towards ICD-11, but it's CMS, Center for Medicaid Services, does not want to move to ICD-11 just yet in this country. So anyway, there's a basic problem. We are doing what we can to try to get specific codes because we think this is going to be important not only for understanding the impact of all of these disorders, it's going to be part of identifying patients for trials and when we want to get trials going for things like L-serine or Radipradil or any of these other things that we think that we want to do in the very near future. So to go over again the functional analysis and what we think, why we think this is important, and we just heard some really great talks in one of the expert sessions about some of the mouse models and how they're coming along. But the underlying premise is this, is that the functional data will advance clinical opportunities. And as you may recall, that what we're trying to do is functionally stratify everybody. So if you're simulating a synaptic current in one of your expression systems where you've expressed one of the mutants, is that what your wild type current will look like is the black line here, but in a loss of function change, it may look like this, or a gain of function, it may look like this. And for a gain of function, it could be bigger, as well as have this change in the time course. And for a loss of function, it could be smaller, as well as have a change in that time course. So it's a combination of those two features that can either push you over the edge to be gain or loss of function. But the question is that we're going to talk about where's that edge. So when things are done in Steve's lab, there are 8 parameters that are assessed to determine the net effect of changes at synapse for and for non-synaptic receptors. And as I said, there's the amplitude, and then there's this time course. And basically, the charge transfer, which is what we think is important, is the amplitude times that time course. And as you all remember, of all the cartoons that we've seen over and over today, is that there's this part that spans the membrane of the protein. Then there's an important part where the glutamate binds, and this other part on the outside that senses what's going on in the synaptic cleft, the N-terminal domain, and then, of course, the pore through the membrane. And then there's a part that we really don't fully assess, which is the C-terminal domain, but it is going to clearly influence things like surface expression, as well as regulation of the channel. And of course, all of these things we think are going to influence things like clinical stratification, such as all of the features that are associated with your GRIN disorder. And one of the things that has been really important to us is to have harmonized data collection throughout the world, partnership with parent groups, and do this in longitudinal study. And this has been really fun to put this all together with Johannes Lemke in Leipzig and Jennifer at Columbia and Simons. So this is that classification of where we're trying to go, is that where is that gray line where you go from gain of function to loss of function, and how sure are we where we're going to put you? And so Steve has put together a nice initial stab at where we think this should be in terms of how we eventually ascribe to you your gain or loss of function. And so there are different categories. There's likely loss of function, possible loss of function, likely gain of function, possible loss of function. So this is that gray area, but it is all meant to be associated with specific changes in those aspects that are assessed in terms of those different features. And basically, you end up with a summary where you're either likely loss or gain, possible loss or gain, no detectable effect. Things can be also often uncertain or complex. And then there is this likely loss of function with the star, which basically means that in the expression systems, it's so hard to get the channels to express themselves that we think that it's probably a loss of function. So it's important, again, I feel like I'm a broken record as I tell everybody over and over again, we really want you to join the registries if you haven't already, because this is how we're going to try to put this all together in the end. And there is a one-stop shop, which is through the GRIN portal. But basically for Dr. Lemke's group, if you're in Europe, Asia, Africa, or other, or if you're North, South America, or Australia, this is where you'll go for this. And again, if you remember that generally gets you where you need to be. So this is what the portal looks like. This is how it directs you where you should go. And so what do we know so far? Now, there's been some nice, several nice papers where Dr. Lemke's group has put things together. And there was a recent review that we went through the different syndromes, but basically to be very, very general, and this is what we put together to describe and make our case to the CDC, is that each syndrome was unique and needed its own code. Was we said that these are things that vary by the syndrome: developmental delay, intellectual disability, autism, speech issues, gross motor, fine motor, seizures or not, or dystonia or not, storms or not, but there are specifics to each syndrome. GRIN1, the symptoms are present in infancy with severe developmental delays and epilepsy. We know this is, again, it was a generalization. This isn't true for everybody. GRIN2B, present in infancy with variable, less severe developmental delays, less frequent epilepsy, sometimes brain malformations. GRIN2A, present with distinct forms of epilepsy and language disorders of childhood. And finally, GRIN2D, present with medically refractory epilepsy in the newborn period, less than a month. And I put this in italics because I think Jen's going to talk to you more about GRIN2Bs. GRID disorders, for a long time, these were thought to be generally autosomal recessive disorders. That is, you have both alleles that are affected, and these were nonsense whole and partial gene deletions. And there was a specific syndrome associated with that. It was called spinocerebellar ataxia type 18. And these were the features associated with that. But now it's been seen that there are missense variants that cause a gain of function within just one of the GRID alleles. And these are emerging phenotypes. GRID1, really there's more to be done here, but for GRID2, there is a phenotype associated with ataxia, cerebellar atrophy, and developmental delay. And there's a paper in process from Steve's group to look at that more. So where are we in terms of collecting things at the University of Colorado? So just to give you where we are is that we've got 52 GRIN1s, 42 GRIN2As, 75 GRIN2Bs, 7 GRIN2Ds, 3 GRIN3Bs, 1 GRID, 2 GRIAs, 1 GRIA2, 3 GRIA3s, and no GRICs yet. This is how they kind of break down in terms of the missense and nonsense. We've got a handful of deletions or duplications, and these are the ones that have functional analysis. Now, of course, there are a lot more functional analyses that are done on patients. Steve, you were saying it's like 650, but these are just the ones that overlap with our group. And thank you. We now have 17 follow-up questionnaires returned. So that's good. Thank you for answering those emails and turning the follow-up questionnaires. And I went ahead and I've swapped my slides around because Liz asked the other day. But basically, if we summarize everybody, basically here, Johannes's group is kicking us with how well he's doing with his awesome recruitment there. But these are our totals. And this is important because now that we have these sorts of numbers here, we think that we have enough to start making these sorts of comparisons about if you have this group with these symptoms and this group with those symptoms, and these are, on the spectrum of gain and loss of function, we can start to make some sort of sense of all of this. So we've beat the goal of, we wanted to have at least 50 in each of these groups. And so we think we're there really to do this for the GRIN1s and 2Bs. There's already been some work with GRIN2As. And so we're going to look at that and see if there's anything else we can add to that. And we're going to carry on doing our smaller studies for the GRIAs, GRIDs, and GRIN2Ds. And in terms of what our database looks like, we've got basically what we do is you all enter the information, but we have to do some data cleaning to make sure that all the records are complete. And there's a physician assessment by myself and Dr. Park for each of the records. And we kind of summarize in terms of what's going on with everybody there. And then of course, we want to combine all of this with our partners and turn it into big papers that suddenly make everything clear to everybody. We hope that it works out that way. So thank you for listening. Thanks for to all you families who've filled in the registry questionnaires. We really appreciate your time and effort. We recognize there is a definitive diagnosis. It's called form fatigue, but we appreciate you filling out the forms. And certainly thanks very much to our funders, CURE, GRIN2B, Simons, and the Children's Hospital Colorado Foundation. So I'll take your questions. Clear as mud. Excellent.
Yeah, thanks. Very nice. I just wondered the prevalence of patients that you showed based on the 2020 data, presumably. Is there a plan to update that based on 2023? Because the numbers that you had in your list didn't seem to quite match with the prevalence from that 2020 study. Is there an updated list? Or?
I'm going to make Johannes answer that question, please, sir. Thank you, sir.
Yeah, hi. So the numbers of the prevalences are estimates. There was a publication from Dennis Lau who calculated the estimates of potential prevalences for all human disorders, actually. And I wrote a commentary on this. I was invited to write a commentary on this article, and I used this algorithm from Dennis to calculate this exactly. What would it look like for GRIN 1 to A to B and so on? So this is hypothetical prevalences. That's not real life world data. So, yeah.
As Jen was murmuring, if we had ICD-10, it would be so much easier to do this. All right, thank you. Oh, no, one more. Go.
Just on that issue, I mean, is that something as a layman that we should be writing to our senator about? I mean, I don't know what the political situation is or how that works at all. You said you've been petitioning for it to the CDC.
Well,
is that something that we need to deal with and think about?
You know, this is one of the reasons why you have CURE GRIN and GRIN2B is that this is, you know, something that Keith and CURE GRI has been working on to. I mean, we've worked with Combined Brain as part of our petition. That's, you know, Keith pulled me in to do the presentation for this, but along with a number of other disorders, when we recently had this phone call with people from the CDC to do this. So, we're, you know, I think that's a good question. Do we need to have this huge, massive pile of signed papers that show up on this person at the CDC's doorstep? I don't know, but we're trying to figure out what's the best way to move them forward. We are having a barrier with the people that we think that should be supporting us in this regard. For some goofy reason, the American Academy of Pediatrics, the American Academy of Neurology, the Child Neurology Society feel that clicking on something and pulling in a drop-down list is too much work for a physician. I mean, I'm overburdened. I've got a lot of work to do, but it's okay, I think, for all of us as physicians to do this and code people correctly. It's just the right thing to do. But we're getting some pushback, unfortunately, from our parent organizations.
Yeah, I just wanted to add on to that with the conversations about the ICD-10. I think it really is important to keep shouting about it. The people who decided, it's just a group of individuals at the CDC, and what they felt is that where there's not clear kind of like actionable treatment plans for these diseases, what does it matter? So you know, I think that we need to make the argument about why it matters and make that really clear.
Yeah, that was part of the discussion that we had on this presentation over a couple of days with them, is to tell them why it mattered, and such that they would realize, you know, it is important. And the arguments that I made to them on the phone, because I was getting pushback from somebody at the American Academy of Pediatrics, who is an emergency room physician. So I put it into his language. I said, you have a patient that comes into the emergency room, they have a GRI disorder, and you want to give them ketamine for the procedure that you're about to do. That is a common thing that they would do. And I tried my best to put it in something that he could understand. And, you know, anyway, we're still where we're at.
Thank you very much, Dr. Benke. So the one thing I'll just add to answer that question is there's a number of us groups, a lot of us through Combined Brain, but also some some associations representing neurological diseases that are working together. And it seems that what we've heard from the CDC is that they're not necessarily in the habit of counting individual letters, but what they want, what would influence them are the two associations that Dr. Benke mentioned, the group of neurologists and pediatricians. And so we may actually be doing together, we may be doing something where we're asking families to reach out to their neurologists and pediatricians to put pressure on the associations. So we presented about 2 weeks ago. We have 2 months of consultation. So we've got about 6 weeks left to make a difference. So we may be reaching out very soon about that. So I encourage you guys all to help.
Once I'm up here, I'm good. Hi, I'm Jennifer Bain. I'm a child neurologist. On behalf of all child neurologists, please don't email us. Email the board of, email AAN and American Academy of Pediatrics. But I've definitely understood the strength of ICD. So I am a child neurologist at Columbia University. I take care of kids with neurodevelopmental disorders and a lot of genetic concerns as well. I'm very animated. I like to pace. I'm not pacing. Yes, it was skiing. Yes, I had surgery a week ago, and I'm so glad that I'm here. I have to say, for those who were in Dr. Trinellis's lecture just before this, he showed a picture from the 2019 group, and I've really had the amazing ability to be with this group in I think 18 and 19 and now 22 in person. And I think what's one of the great things about being where I've been is that I also have been working in collaborations with Simon Searchlight. So I'm going to be presenting some data that's from Simon Searchlight, but this is actually your data. So when you met all those wonderful people outside from Simon Searchlight in the corner, and they will be here after this for those who have not, and they are available, I'll give you their information. This is the information that you've shared back. And the really key thing is that harmonization really has been occurring. So since we did sit down in the lobby in 2019 in Atlanta, we actually did sit down and say, we don't want people to have to do 8 million forms. How can we align so that we're actually gathering the same data? And it's actually happening, guys. So it's very exciting. I didn't ask how to use this. Yeah, okay. So I'm going to take off my Columbia neurology hat and I'm going to put on my Simon Searchlight hat. And I'm going to present a little bit more in detail kind of about what the goals are. Where does this go? There, Simon Searchlight. So Simon Searchlight is an international research program. And the goal of it is to accelerate science and improve the lives of families affected by rare genetic neurodevelopmental disorders. It's a great big team up here. The PI is Wendy Chung up in the top, and many of the members that have been here this week are seen here. What are the goals of Simon Searchlight? Well, the mission really has been to shed light on neurogenetic disorders. It's not autism only, it's actually neurogenetic. So it's genetics first. And the goal is really to collect high quality data, natural history, meaning longitudinally, and then really be able to foster those relationships between the researchers and industries and families. What they do, for those who have not been introduced to Simon Searchlight, is collect detailed medical information, behavioral histories from validated measures that maybe you've actually encountered at school and psychologists. They also collect and facilitate biospecimen collection. I take all that information, we put it all together. We have some amazing team members that put it back, and then guess what? We share it back. And so that's what we're going to do today. And I'm really thrilled because when I first presented to the GRIN 2B group, we didn't have the numbers we have today. And I really hope that I can keep coming back here, and the numbers that we're seeing grow from GRIN 2B will start kind of happening with your other groups as well. The data is freely shareable with qualified researchers and the samples as well. And so the goal really is to connect these research participants and the researchers and get us all in the same room so we can talk about paroxysmal sympathetic hyperactivity and the models that we have available in your toolkit that you guys are contributing to. And obviously the goal of this is really to promote better understanding of all these genetic changes. So Simons Foundation is kind of the overarching. Safari is the Simons Foundation for Autism Research Initiative. There are different parts, different projects, but they're kind of all related in a continuum. So on the left, you can see we have a project that's the newest. It's called Spark. This comes from the diagnosis first. If you have a diagnosis of autism, you can participate and have genetic testing from that perspective. If they find a genetic reason that's contributing to your autism risk, then you get funneled into Simons Searchlight. This is a genes first approach. So you don't have to have an autism diagnosis. We're talking about neurodevelopmental phenotypes, intellectual disability, global developmental delay, epilepsy, etc. And so these are individuals who have a genetic diagnosis first, and we're trying to go back and figure out how does the phenotype kind of match up. Again, as I mentioned, we'll collect behavioral information over time, clinical data, blood specimens, and we can provide more tools to the toolkit to studying this. And the more recent push has really been, as we've talked about, how can we think about the future, you know, the good, the bad, and the ugly, thinking about when we do lose our family members and we do still want to contribute to the field, the autism brain net really is something we are asking families to think about. It's a hard decision to obviously go through and have that discussion, but certainly talking about it before, I think, can be helpful to hopefully opening up a lot more information to the brain. We do, from Simons Foundation, offer funding for outside researchers. As you've heard, many of the people here have had funding from Simons Foundation. And as mentioned, they really are trying to build that resource, that data bank, so that we can share the animal models, use the Safari gene and the Safari base, and really try to foster that research community. Big green button. As mentioned, Autism Brain Net, this is the program of Safari to really build upon postmortem brain tissue. And donor families are really discussed and talk through these discussions because these are very obviously difficult discussion, and we have more information about that if you're interested. So let's get down to the data. Again, this is between hearing about this and dinner. So we're going to go with GRIN2B first, because this is where the majority of the data that we have so far. So as of March 8th, what we're going to do is you're going to start on the left here. Step one is you sign up online. Isn't that easy? So you don't even have to go anywhere, which is one of the great strengths of Simons Searchlight. You provide your genetic lab report. We want to make sure that people who are participating really actually do have the genetic disorder. It will be reviewed by your genetic counselor team. And then you share your important medical information. Surveys are provided initially and then as longitudinally as well. You have the opportunity, if you so please, to provide a blood sample if you're interested, and we'll talk about some of the people who have done that. And then really, again, what happens when our kid grows up? Well, that's the point of doing something like this. We really want to know and have that information as people are getting older. Simons Searchlight's now 11 years, I believe. Are we at 11? 11? Yeah, 11 years. So, you know, we're getting a lot of data longitudinally, which is really helpful. And so if we look at GRIN2B, clap. GRIN2B, awesome data. This is really amazing, right? So when I first came here, these were like just in the double digits. So 137 have signed up, 105 have confirmed their genetic reports, 84 have shared their medical history, standardized medical histories, 95 have filled out surveys. We have 30 who have provided samples. So this is really great. This is a lot of strength in numbers. And I think the goal is to take this and combine it with all these other great registries and really kind of not double up on the work you have to do, but really kind of gain some information that we can contribute to the field as a whole. So here's where we are with the others, okay? So GRIN1A is the top here. So we have 15 individuals, GRIN2A, we have 18 individuals, and 2D is 3. And I think the GRICs we got to work on, all right? So keep doing it. I'm telling you, if I come back in a couple of years, I want all those numbers all up in the hundreds, okay? We need to show the numbers. And this is probably going to give us more information since we don't have ICD-9 and 10 and 11 coding. What do we collect? Well, at minimum, we're really aiming, we need a lab result to say what you have. And then we really need medical history kind of information about your clinical picture, your clinical phenotype. And then we do take information such as medications. And then there's a whole slew of other online surveys that we're really trying to kind of, we allow everybody the opportunity to participate in them as much or as little as you're able to. And then specific kind of questions or questionnaires could be pushed out depending on what the community and researchers might be interested in. Coming in 2023, there are some newer assessments that will be coming out, such as the ORCA, which is a communication measure that's more commonly being researched right now, behavior problems, more developmental milestones, using different standardized scales such as the PDCAT or Pediatric Evaluation of Disability Inventory. And there's been a really great interest in collecting all the EEG raw data. And so, you know, I think you can think about all these great things that you can contribute. You've already had these things done, and how can we contribute that back? So this is what a participant dashboard looks like. And so it's pretty simple. It shows a check mark when you did something. So you get registered, you consent. Everybody in the family can consent if they want to participate. We're not enrolling just the child, we're enrolling the family or whoever is interested and available. And it kind of goes through exactly what you need to do. There's a list of tasks that we can offer for you in your spare time. And then there's some great things like Amazon gift cards after you complete some of your surveys and rewards. And so the goal really is to kind of give back and thank you and appreciate you. But again, you're giving back to yourself and your research community. And so you really, hopefully, and we take feedback. So if you say this doesn't work or this doesn't look great on my platform, let us know. And so what are some of the biospecimens we have now? Well, there are 29 participants who have participated in the biospecimen collection. And so there are currently 12 patient-derived induced pluripotent stem cells that are available. We have 8 cell lines, 16 whole blood DNA sets, and then some saliva samples as well. And these are great. They're readily available for researchers who are qualified research. They're very nominal fees, really mostly for shipping. And anybody who's interested in talking about this, I'm happy to. These are the available iPSC lines, and I believe most of them are missense variants, but there's a few truncating variants as well. Okay, in terms of what types of mutations we're talking about are pathogenic variants. So typically they will collect all information. So remember when maybe some of you had a variant of unclear significance and then it became pathogenic or likely pathogenic? We know that these GRINs are growing and we're learning. So we will enroll you, even if it's just a variant, and then we're gonna continue to follow you. And if it ends up being updated later, great, we'll include that in your information. The majority are missense variants, as you can see here. So here's the gene, here's like a nice little schematic, and each one of these lollipops represents a person at a specific location. I like to call them typos, right? So the red ones are typos, and then up here is the specific locations of each of those variants. I'm not going to go through them. There are a few frameshift nonsense variants and some splice site variants and some deletions that were not shown here. If you want more information on this, we're happy to share that. In terms of medical data, so here's where we come into the powerful information. You know, we really aim to kind of collect high quality information based on caregiver report. This is a really great slide. It shows that, you know, in the individuals that we have, 72 individuals between 1 year and 35 years, this is just a snapshot at those individuals. Importantly, 66 of those were younger than 18. And so you can see right here, whoopsies, back. Right here, this is showing the abundance of people. So you have more people who are younger, less people who are older. This is so important, right? So we know that there's people who are alive who have this disorder. What we need to do is keep all you guys involved and keep moving forward together, right? Because we need to understand what's happening over time. This is just a snapshot. The average age of people in that 72 people is 8 years old. So as you can see, the majority are younger. So stick with us, all right? Because this is really only going to help the community understand what happens over time. In terms of developmental and behavioral conditions, as are neurodevelopmental disorders, they have language delays, language impairment in the majority. There are about 30% of individuals do present with an autism diagnosis. Anxiety and ADHD symptoms are also common, as well as OCD. Neurologically, I'm a neurologist, so many children get diagnosed with hypotonia or low muscle tone, and then we collect information on other different diagnoses that you might see with your loved one. Coordination problems for sure, cortical visual impairment as well. Seizures are not big, you know, and I think we've shown this within the GRIN2B community. It's not a large epilepsy phenotype, but there are different types of seizures that are seen in these groups. So 72 individuals provided information about this. And in terms of looking at those 30 people who did provide information a little bit more about their baseline seizure, the seizure onset was anywhere from the first month of life to 7 years of age. The average age of first seizure in this group was 2 years of age. And about half of them ended up having to take medication to control their seizures. And about 3/4 still do continue to take medication for their seizures. 17%, meaning 5 of these individuals had a seizure. They actually were able to control them. And it did take about 4 years for that to happen. And it could take a little bit longer. There are vision common problems that we see across the group, such as strabismus or cross-eyed or lazy eyes. And then there's additional medical issues, and neurologic, I think, gets lumped often. If it's not something else in the body, just blame it on the brain. What we have to remember though, is that very often your pediatrician just has to remember that you're still a child and you can still have a earache and a strep throat and all those other things that other children can have as well, or adults. We do collect information about medication, and the goal of this is really, on your forums, you're probably talking on Facebook and other social media to really understand what medications might work for you that might not work for somebody else. And this can be really helpful. And so, I think the majority of the medications that we're seeing is related to sleep and gastrointestinal issues. So this is very, I think, helpful information overall. What I did want to say, there actually is no particular medication that seems to be helpful for seizures. And I think we've learned why, right? Because some of the variants cause too much activation and some cause too little. And so overall, there's not really one specific medication that's perfect. The goal of this is to harmonize our data sets and then maybe say, is this medication working better for these functional groups, et cetera. The Vineland Adaptive Behavior Scale is something that's, it's a long kind of tedious questionnaire, but it's very helpful. It's a caregiver report that gives information about the adaptive skills. Now, I'm not going to go too much into this because I think the graphs can get a little bit tricky to understand. I know it's really busy, but it's actually quite, it's quite helpful. I think people understand where their children are. And the goal really is to provide information back about, well, what's going to happen as people get older. So this is what we call cross-sectional. So if we looked at all the individuals, so here we're looking at 45 individuals with GRIN2B at this point in time. And so each one of these little blue dots is a person with GRIN2B. On the bottom is their actual age. So this person is actually 17 years old. These people are actually 1 or 2 years old. On the y-axis is kind of their level of functioning. So if I actually did the psychological adaptive testing, where would they kind of lie out? And so you would expect, I think let's use 10 as an example. So at 10 years of age, we would kind of expect them to be at that 10 point, all right? And if we draw a line of all the individuals with GRIN2B, we see that they're behind where they're supposed to be. And we know that because that's kind of how we got here. But people do want to know what's going to happen to my child as they get older. Now, this is cross-sectional, meaning I'm just looking at them now. I haven't taken those people and watched them over time. That's the goal, hopefully, of all of these data sets, is to follow them over time. Yes. Oh, that's my 5-minute mark. I think I have like 2 more slides, so we're good. So for example, I'll just give some nice little highlights. So this is a child who's 4 years old, and at the 4 years age, they're about 2.5 years of age in terms of their level of speech, okay? And this dotted line down here is kind of that at around 1 year old, we typically expect somebody to have single words. Does that make sense for everybody? We'll kind of go through a few of these. This is looking at personal care skills. So how well do individuals take care of themselves? Same situation where the blue dots each represent a person. On the bottom is their actual age. On the y-axis up here is their level of functioning age. So the first dotted line down here around 2 is a typical milestone that we would see, is that somebody can feed themselves, not nicely, but they can feed themselves with a fork and a spoon, right? And then at 4 years of age, we think that that child should be able to kind of dress themselves. So what we're seeing here is that typically around 6 years old, we're more kind of seeing, if we look at the group as a whole, they're able to kind of feed themselves. Up here, typically around that 16, 17-year-old, we're starting to see skills that are higher, such as dressing themselves, okay? This is expressive language development. So this is looking at the speech. We just did that. going the wrong direction, personal care, and then social development. Okay, so I think this is the last one. And so similar trend, and you guys are becoming experts in this, the actual age and their level of functioning. So the bottom dotted line is an example of making eye contact. And then the 2-year-old mark is more showing interest in friends. And so for here, again, you have a lower kind of age. So around that 6 years of age is starting when we're starting to make those eye contact and engage in those social relationships. Gross motor skills is a little bit stronger. And so the bottom line is when we're walking without help, typically that's around 1-year-old. In the GRIN 2B group, we're seeing that happen more around 3. In terms of hopping and jumping, we typically see that around 3. And in this group, we're seeing that more around 11 or 12, okay? So that's just an example of the kind of data that they're collecting and kind of at a cross snapshot. And then hopefully as they're getting older, we're kind of going to be able to make some more real longitudinal comparisons between the groups. The CBCL is another checklist that we look at different types of behaviors. And just in summary, for the younger kiddos, so this is 28 individuals who are 1.5 to 5, really some of the noted behavioral concerns or challenges are hyperactive behaviors, chewing on non-food items, we sometimes call that pica, desiring frequent attention-seeking behavior, and difficulty waiting a turn. And then in the older individuals, in the 16 to 18-year-old, we're talking more about frequently being distracted, some obsessions, more frequent attention, and kind of hyperactive behaviors. And so in summary, and I promise we're done, some of the more common issues that we're seeing reported by caregivers are low muscle tone or hypotonia, language delays, eye problems like strabismus, and GI issues. And some of the other issues that certainly is being noted in the group is autism, other vision problems, seizures, scoliosis, and attention deficit problems such as ADHD. So thank you very much. Again, I want to say clap for you guys because the GRIN 2B group did that. And I implore you to keep moving those data sets up. Questions?
Question through the app. So either someone in the room or someone watching virtually. Actually, 2 questions that are similar. So I'm having trouble finding it. But one is okay, is Simon's going to expand to GRIA, GRIK, and GRID patients? And if a GRIA patient has an autism diagnosis, does that mean they can donate to Spark, but not Simon Searchlight?
I will answer number 1 first. So number 1 is, will they expand to other gene groups? Yeah, so the goal is to expand to as many of the GRIs as possible now that we recognize this as a gene group. The way it works logistically, whenever anybody gets involved in human subject research, there is ethics. There's an ethical board. You might have heard the term Institutional Review Board or IRB. For this project, typically we need at least 5 people to kind of be in a group before we kind of release that information out. So there might be some people, those 1 or 2s, but we can't release that information out yet. Obviously, if you have one parent who has, whatever disorder, disorder X, and they say, oh, everybody signed up for Simon Searchlight, I did, and we know there's one person, that's that person. So the goal really is to try to keep it as de-identified as possible. So let's do the lemming thing and everybody do it together. And the more people you get, the more that data will kind of come back. The second question was about
if someone has
blood to Spark. I'll get back to you on that one. I believe if you have the autism diagnosis, you can go in and get that. If you already have the genetic testing, I'm not sure what whether you would be enrolled or not, but I can find out. Okay.
And the other thing that I've heard from Simon's, I think there's a meeting in about 2 months where they're considering new genes to add. And so the GRIAs and GRICs are on the list.
Yeah. And I think, what we've recognized is that we're talking about gene groups. How do we build numbers? We're going to all boats rise. So, we started with GRIN 2B. They did a great job kind of pushing through forward with this collaboration. And I think hopefully the other GRINs can jump on board too and kind of move forward. And I think if you continue to show the need that we are a large gene group and we, work together, that power will be seen much more.
In terms of medical history, do you guys collect CBC and urinalysis type of data as well?
What was the first? I heard urine.
complete blood cell count?
Oh, CBCs. We don't collect the blood counts per se, no. You know, we don't do the blood testing directly itself. We don't do like the urinalysis or anything like that. There are, like I said, like they'll collect the biospecimens. It sits in a bank. They're not doing the research on it. They're making that tool available to other people. And I think, you know, people probably could donate information of testing that they've had, but they're not doing the actual testing itself.
I think you guys should encourage people to donate those kinds of results, because there could be things that match, like a lot of people have in common that could pop up. Like, for example, I thought it was interesting that a lot of people take allergy medicine, and GRIN2B is very important for neutrophil function. So perhaps there could be some discrepancies within the CBC that we should be looking for.
That's exactly right. I mean, I think the goal of this is to kind of kick baseline information for everybody. And then if there's researchers who are particularly interested in neutrophil function, you know, go in there and say, okay, now I want to get all the clinical phenotype data I can there, and really kind of deeply dissect the data that they have there. Thank you.
Thank you.
Hi.
Thanks. Hi, Jennifer. How are you? Sorry. Sylvia is my student, and we have a similar question, which is when patients sign up, is there any kind of consent to release for research, consent to release de-identified medical information for research purposes? Because we would be very interested if we see something in our animals, is that true in the patients? And, you know, we have a feeling that people might have submitted their, you know, when they go to the hospital, they probably run some of these standard tests. And, you know, it would be nice if we could mine some of that information. Absolutely. Without having an extra blood draw for it.
100%. So we can definitely talk a lot offline about the specifics, but this is exactly the goal. The goal of Simons is not to do the research itself. It's to facilitate, to build the toolkit. Okay. So they collect all the information, and then someone once told me it's like, you know, the library, right? So you can borrow that information. You can borrow that information. You can borrow that information. So when somebody consents to being a part of Simons Searchlight, they are consenting to provide all of their data de-identified to be shared. So that's the exact purpose of the project. And so we can definitely talk offline about seeing what information is being collected from your group. Mm-hmm. All right, thanks so much. Enjoy dinner.
Wrapping things up, we have a virtual presentation from Dr. Allan Bayat on GRIAs.
Hello, everyone. My name is Allan, and I'm a medical doctor from Denmark. I'm here because I want to give you an insight about the kind of research we do in my team, which is actually about all GRIA-related disorders. So the talk today is going to focus on GRIA 3, but I mean, it could also just basically have been any of the other GRIA genes. So we want to help people with GRIA related disorders to better understand their disease and help them to get a better treatment. So basically, that's what we want to do. Before we go further, I just want to give you a short introduction to this very important and really cool system in the brain of how cells interact with each other. So here on the picture, we have two brain cells. And the first brain cell in yellow creates a current. So cells talk with each other by creating an electrical current. So the current starts in the cells, and then it reaches the end. And then what happens then is that a substance called glutamate is then released. And glutamate is really important. So remember that name. Glutamate is really cool and binds to two receptors on top of cells around the first cell. So these receptors are called the NMDA, which is encoded by the GRIN genes, or the AMPA, the AMPA receptor, encoded by the GRIA genes. And what happens when glutamate binds? Well, then the receptor opens an electrical current is then created, and that turns on the next cell, which then passes the electrical current, and then more glytamate is released and more cells are activated. So this is really an important system in the brain. Now we're zooming into the AMPA receptor. The AMPA receptor is built up by small subunits or building blocks. And these building blocks are created by the GRIA genes. So we have 4 GRIA genes, GRIA 1, 2, 3, and of course 4. They're all important and all very cool, but GRIA 3 is very unique because it's X-linked. And what does that mean? Well, when you talk about X-linked disorders, you commonly think of something where healthy females are carriers of a disease and then pass this on to, for example, boys that get ill. So boys are ill because they only have 1 X chromosome, while females are carriers because they have 2 X chromosomes. The question is, why is research in GRIA important? I think it's important because we have so limited knowledge. And that's because very few patients and very little research has been done in human diseases related to GRIA. So we just need to do better. And how to overcome this? Well, we have envisioned a project where we start collecting a lot of clinical information, preferably as much as possible, from patients that have or could have a GRIA-related disorder. So all of this information is collected in our registry so we can understand what kind of symptoms you're facing and also how symptoms evolve over time. Then we start studying the mutations, and we do this here in the lab. And what we do is we ask 3 questions. First question is, does this human mutation affect the function of the AMPA receptor? And then we study this. We can measure if a mutation is making the AMPA receptor work poorly or differently. Next question is, well, if it's affecting the AMPA receptor, how is the effect? Is the AMPA receptor too active, which we call a gain of function, or is it not functioning enough and has a lost effect? This is a loss of function. So we can have either a gain of function or a loss of function mutation. And the third question is, how can we reverse these effects? So if we have a gain of function mutation, can we, for example, use a drug called perampanel, which is a blocker of these AMPA receptors, to sort of improve the health and symptoms in GRIA patients? And in the end, all of this is passed on to you or your medical doctors for better counseling and treatment. So let's look a little bit further into what we're doing in the lab. So we work with frogs and with egg cells from frog. Egg cells are called oocytes, and we harvest these oocytes from the frogs, and we use this to study the human mutations. So you see here, we've harvested these cells from the frog, and they look like small, tiny balls here under the microscope. Now, these cells actually do not express the AMPA receptor, so we need to make them express this. We do this by injecting them with a small copy of the human gene. Let's say we want to study GRIA3, so we inject them with a copy of GRIA3. And this can be a normal, healthy copy of GRIA3, or it can be a copy of GRIA3 that has a human mutation that we want to study. So in the figure, you can see I'm injecting it with a copy of GRIA, and it has a human mutation. Then I'll let the oocytes rest for a few days in the fridge. And what happens is that the oocyte will start building these building blocks for GRIA, because now it sort of has the recipe for GRIA. It starts building these subunits and making AMPA receptors on top of the cell. After 3 or 4 days, we take out the oocytes, put it here in a barrel, we stick 2 needles within it, and then we pour different chemicals on top of it, and we can then measure how the AMPA receptor is functioning, which is pretty cool and fairly easy to do. Every time you do research, you need a control. So here we've taken an oocyte, we've injected it with the normal functioning copy of, let's say, GRIA3. So the AMPA receptors here in the cell are working just as they should in healthy patients. And what happens is that we add increasing concentration of glytamate. As you see, the numbers are increasing, meaning that you're pouring more and more glytamate on top of the oocyte. And as you remember, glytamate activates the receptor, it opens it, and you see that the more glytamate you pour, the bigger current you get. So this is like the control, and we compare it with other results from the human mutations, and I'll show you this later. So let's get back to the patients. So we have at least 25 patients with GRIA-related disorders, 14 males and 11 females. And this is mind-blowing because now we're showing that a lot of females can actually be affected. That's really cool, I think. And of course, also important for counseling. The majority of patients are, of course, children. So the average age is about 10. Some are much younger and some are older. As you see, there's some adults in their 30s. All have global developmental problems, but it ranges from mild to severe. So let's have a look at the mild spectrum. I've listed some of the symptoms here. Often the early milestones are normal, but then around infancy, you start seeing problems with development. Often patients learn to walk, but the gait is unsteady. Some have epilepsy, most do not, but in terms of treatment of seizures, it's quite easy and patients do become seizure-free. This is the worst part of the spectrum, where patients unfortunately are affected immediately at birth. They're very stiff and cannot relax their muscles, so that really hinders development. They cannot feed and are depending on a feeding tube. They have no or limited eye contact. They don't really develop and have epilepsy starting very, very early in life, and it's really difficult to treat it. Here I'm showing you 2 girls that have mutations in GRIA causing the AMPA receptor to be very overactive. So 2 gain-of-function mutations. The small baby has a strong gain-of-function, and the bigger girl, the infant, has a less strong. And what you can appreciate is how stiff they are. You can see that the baby has a feeding tube. You can see how stiff she is. She's unable to relax her arms and her legs. Noise and even touch makes her twitch. I'm going to fast forward just a little bit. So again, you can see how incredibly stiff she is. This young girl over here also has stiffness. She's a bit older, but you can see her eye contact is actually quite good. And she's trying to interact. But she also is very stiff in the arms, legs, neck, and the trunk. Patients often have behavioral or psychiatric problems. In terms of behavior, it can be that they can be very sensitive, very anxious, but often those with loss of function mutations are aggressive and have a short temper. But across all patients with gain of function or loss of function mutations, being anxious, being sensitive, being hyperactive, having too little attention span, or having problems with social interaction is seen and is quite common. So as you remember, we had 25 patients. in these 25 patients, we found 17 unique mutations. So I'm showing you 3 examples of results from 3 different people. So in the box, that was the control. Above here, you have 1 patient that had a mutation on position 609. This mutation is not really causing any change in the current. And of course, we did a lot of other tests. And in the end, we went back to the clinician and to the family and said, we do not think this mutation is the cause of illness, and we suggest maybe looking for another cause by going back to the genetics again. In the middle, we have a patient with a gain-of-function mutation on position 654, where you see that the currents are much stronger, while down here, this is a loss-of-function mutation on position 776, where there's hardly any current at all. So examples of a gain-of-function or a loss-of-function mutation. Now, the cool stuff is that we can merge these lab results with the clinical data we're starting to see patterns of symptoms. So let's say, for example, in both gain or loss of function mutations, patients do have epilepsy, but we see that if you have a gain of function mutation, you do have a tendency to start seizuring much, much early in life, often around the first months of life. Whereas if you have loss of function mutation, the seizures often start after your first birthday. We also see that the stiffness and the startling and hyperexplexia, as you saw in the baby, is something that we almost only see in gain-of-function mutations. So that's like what we call clinical biomarkers of whether something is a gain-of-function or a loss-of-function mutation. The take-home message is that both males and females can be affected. Males are often carrying mutation that they have from their mothers, and they often have mutations that are loss of function, while females often are the first people in the family to have the mutation. The mutation is not inherited from the parents, and in females often is caused by gain of function. And then I just very briefly want to thank GRIN and families and people around the world that have helped us with our research. And I just have one final comment, which is that we would really like to get in contact with families and patients that have the symptoms that you saw on the video. So this stiffness and involuntary movements, we really want to study that further. So if you are a parent or a caregiver of a patient that has these symptoms, then please reach out to me. We'd like to study some videos and understand what kind of symptoms patients are having and how they develop over time. We really think that's crucial and important aspect as well. And with that, I would like to thank you all, thank you the audience, and wish you a very, very nice conference. Thank you.
So thank you to Dr. Benke, Dr. Bain, and Dr. Bayat, all B's, for your presentations today. And so we're at the end. I think they're gonna put up a couple final slides. Did you guys have a good time? Did you learn stuff? Was this worthwhile? Good. Oh, it's okay, because the other session's coming in now. We're just waiting for the slides. You guys are gonna have high expectations now, but these are not great slides. I'm just telling you, like, what we're waiting for maybe isn't worth it. Okay, okay. It's okay. We'll go without the slides. I did want to put up the sponsor slide again, which we have here. So just want to thank all of the sponsors GRIN Therapeutics, Simon Searchlight, Safety Sleeper, Pneumora, Cure Epilepsy, Sage Therapeutics, Ovid Therapeutics, Homology Medicines, GRIN2B Foundation, Children's Hospital Colorado, as well as our 4 family sponsors, Carter Williams, Metric Family, Team Bretman, and Downing Family. Was there one before that? Okay, yeah, so if you would like, if you are going to the Science Center tomorrow, just a reminder, is this time good? Meeting, yeah, so Lauren and Casey will meet you in the hotel lobby tomorrow at 8:45, or you can make your way over later. So after that, slide with the sponsors. Okay, yeah, next one, please. Okay, so want to just make sure we thank some people who have helped make this weekend possible. So first I want to thank the whole GRIN team. So Denise Renner, Megan Collins, Lauren Williams, Brittany Brown, and the rest of the board of directors, the 3 who are here today and the others who couldn't make it. Thank you so much. From the MedLogix team, Jennifer Hernandez, who's here, and Lindsay Luce and Sue Sessa, who aren't here today, but have just been great in helping us put together this amazing conference. Thank you to all of you who spoke. We had a lot of great presentations. Thank you to the AV team. Thank you to the hotel staff. So let's give them a round of applause. And finally, I just want to call up 2 people in particular. So if Lauren and Megan could come up for a moment. So as many of you know, we had our awards dinner last night, and 2 of the people who got the most nominations were Megan and Lauren. But the selection committee decided that we didn't feel it was appropriate to be giving awards within GRIN while people are employees of GRIN. So they have to wait. I know that they'll get nominations when they're not here anymore, but it's clear from the nominations and what you guys wrote about Megan and Lauren, how much the community really appreciates the work that you guys do. So I want to thank you, and I'm sure everybody here wants to thank you.